Spiders. For some, the word itself is enough to send people into a state of shrieking fear. In this edition of Doc TV's monster horror documentary series, we'll be weaving a web of truth around these incredible creatures. What are spiders? Spiders are oxygen-breathing arthropods that bear 8 legs, chelicerae with fangs typically able to inject venom, and spinnerets that produce silk. They are the most significant order of arachnids and rank 7th in total species variety among all hierarchies of organisms. Spiders are found globally on every mainland except for Antarctica and have become based in nearly every land environment. As of August 2022, 50,356 spider species in 132 families have been documented by taxonomists. However, there has been controversy among scientists regarding how families should be organized, with over 20 classifications suggested since 1900. Unlike insects, spiders don't have antennas. In all but the most primitive class, the Mesothelae, spiders have the most centralized nervous systems of all arthropods. All their ganglia are fused into one mass in the cephalothorax. Unlike most arthropods, spiders bear no extensor muscles in their limbs and extend them using hydraulic pressure. Their abdomens have appendages modified into spinnerets that create silk from up to 6 types of glands. Spider webs range widely in size, shape, and amount of sticky thread used. It now seems that the spiral or web may be one of the first forms, and spiders that make tangled cobwebs are more plentiful and mixed than all weaver spiders. Spider-like arachnids with silk-producing spigots emerged in the Devonian period about 386 million years ago, but these animals lacked spinnerets. True spiders have been discovered in Carboniferous rocks from 318 to 299 million years ago. They are comparable to the earliest surviving suborder, the Mesothelae. The main classes of modern spiders, Mygalomorphae and Araneomorphae, first emerged in the Triassic period, 200 million years ago. The species Bachyera kiplingi was portrayed as herbivorous in 2008. Still, all other known types are predators, primarily preying on insects and other spiders. A few larger types also prey upon lizards and birds. It is believed that the world's 25 million tons of spiders kill 400 to 800 million tons of prey annually. Spiders use a wide range of strategies to capture prey, trapping it in sticky webs, lassoing it with sticky bolas, mimicking the prey to avoid detection, running it down. Most catch prey mostly by sensing vibrations, but the active hunters have sharp vision, and hunters of the genus Portia display signs of intellect in their tactics and proficiency to create new ones. Spiders' innards are too thin to handle solids, so they liquefy their meals by flooding them with digestive enzymes. They also gnaw food with the bases of their pedipalps, as arachnids don't possess the mandibles that insects and crustaceans have. To avoid being consumed by the females, which are typically much bigger, male spiders identify themselves as potential mates through various complex courtship rituals. Males of most species endure a few matings, restricted mainly by their short life cycles. Females weave silk egg cases, which may house hundreds of eggs. Females of many species care for their young by hauling them about or disseminating food with them. A minority of species are social, building collaborative webs that may house anywhere from a few to 50,000 individuals. Social behavior varies from unstable patience, as in the widow spiders, to collective hunting and prey sharing. Although most spiders survive for about 2 years, tarantulas and other megalomorph spiders can live up to 25 years in captivity. While the venom of several species is harmful to humans, scientists are now exploring the usage of spider venom in treatment and as non-polluting pesticides. Spider silk delivers a combination of lightness, strength, and elasticity that is incomparable to synthetic materials, and spider silk genes have been inserted into mammals and plants to see if these can be utilized as silk manufacturers. As an effect of their wide range of mannerisms, spiders have evolved into standard symbols in art and mythology, representing diverse kinds of tolerance, brutality, and innovative capabilities. An unreasonable dread of spiders is known as arachnophobia. We'll get more into that later on. Spider anatomy. Spiders are chelicerates and thus arthropods. They have segmented bodies with jointed limbs covered in a cuticle made of proteins and chitin. A spider head comprises several segments that connect during the embryo's development. Their bodies consist of two tagmata, sets of segments that perform comparable functions. The foremost one, called the cephalothorax or prosoma, is a total fusion of the segments that in an insect would create two separate tagmata, the head and the thorax. The rear tagma is known as the stomach, the abdomen or opisthosoma. The cephalothorax and gut are linked in spiders by a small cylindrical section, the pedicel. The design of segment fusion that forms chelicerates' heads is unique among arthropods. The first head segment vanishes at an early stage of development, so chelicerates lack the antennas which most arthropods do. Chelicerates' only appendages ahead of the mouth are a pair of chelicera, and they don't have anything that would serve as jaws. The first appendages behind the mouth are called pedipalps and perform various functions within different groups of chelicerates. A spider's cephalothorax and the abdomen are bound by a small cylindrical pedicel, allowing the stomach to move independently when producing silk. The upper surface of the cephalothorax is covered by a single convex carapace, while two relatively flat plates cover the underside. The abdomen is egg-shaped and soft. It exhibits no sign of segmentation, except that the early Mesothelae, whose living members are the Lithistida, have segmented plates on the upper surface. Circulation and respiration. Spiders are coelomates. The coelom is constrained to remote areas around the excretory and reproductive systems. Its place is taken mainly by a hemicoel, a cavity that runs most of the measurement of the body and through which blood circulates. The heart is a chute in the upper part of the body, with a few ostia that act as non-return valves, permitting blood to enter the heart from the hemicoel, but stop it from leaving before it contacts the front end. However, in spiders, it inhabits only the upper part of the abdomen, and blood is released into the hemicoel by one artery that opens at the rear end of the stomach, and by branching routes that pass through the pedicle and open into several parts of the cephalothorax. Therefore, spiders have open circulatory systems. The blood of many spiders with book lungs includes the respiratory pigment hemocyanin to push oxygen transport more efficiently. Spiders have evolved several different respiratory anatomies based on book lungs, a tracheal system, or both. Mesothelae and megalomorph spiders have two pairs of book lungs filled with hemonymph, where openings on the ventral surface of the abdomen permit air to enter and diffuse oxygen. This is also the case for some basal araneomorph spiders, like the family Hypochilidae. Nevertheless, the remaining members of this group have just the anterior pair of book lungs unchanged. In contrast, the posterior pair of breathing organs are partly or wholly modified into tracheae, through which air is diffused into the hemolymph or straight to the organs and tissue. The tracheal system has most likely evolved in small ancestors to help resist desiccation. The trachea was initially connected to the surroundings through a pair of openings called spiracles. Still, in most spiders, this pair of spiracles has merged into a single one in the middle and moved backward near the spinnerets. Spiders that have tracheae have higher metabolic rates and better water conservation. Spiders are ectotherms, so environmental temperatures impact their activity, feeding, digestion, and excretion. Uniquely among chelicerates, the final part of spiders' chelicera are fangs. The great bulk of spiders can utilize them to inject venom into prey from venom glands housed in the roots of the chelicera. Uloborida, Holarchida, and some Lithistida spiders have lost their venom glands and slay their game with silk instead. Like most arachnids, including scorpions, spiders have a slim gut that can only manage liquid food and 2 sets of filters to control solids by keeping them out. They use one of 2 other systems of external digestion. Some inject digestive enzymes from the midgut into the quarry and then suck the liquefied tissues of the target into the abdomen, eventually leaving behind the empty husk of the prey. Others gnaw the game to a pulp using the chelicera and the bases of the pedipalps while bombarding it with enzymes. In these species, the chelicera and the bases of the pedipalps create a pleural cavity that harbors the food they are processing. The stomach in the cephalothorax serves as a pump that transmits the food deeper into the digestive system. The midgut handles many digestive ceca, chambers with no other exit, that extract nutrients from the meals. Most are in the abdomen, which is monopolized by the digestive system, but a few are located in the cephalothorax. Most spiders recycle nitrogenous waste products into uric acid, which can be discharged as a dry material. Malpighian tubules, also known as little tubes, remove these debris from the blood in the hemicoel and discard them into the cloacal chamber, from which they are ejected through the anus. Production of uric acid and its removal via Malpighian tubules are a water-saving feature that has developed independently in several arthropod lineages that can exist far away from water. For example, the tubules of arachnids and insects develop from entirely separate parts of the embryo. Regardless, a few primitive spiders, the suborder Mesophilae and Infrorder Megalomorphae, maintain the ancestral arthropod known as the little kidneys, which utilize considerable amounts of water to excrete nitrogenous waste derivatives like ammonia. Central nervous system. The primary arthropod central nervous system consists of a pair of nerve cords operating below the gut, with paired ganglia as local command centers in all segments. A brain formed by fusion of the ganglia for the head parts behind and ahead of the mouth, so that this conglomeration of ganglia surrounds the esophagus. Except for the primitive Mesophilae, of which the Lithistida are the only surviving family, spiders have the much more centralized nervous system that is characteristic of arachnids. All the ganglia of all components behind the esophagus are connected so that the cephalothorax is primarily filled with nervous tissue, and there are no ganglia in the stomach. In the Mesophilae, the ganglia of the gut and the rear part of the cephalothorax stay unfused. Despite the relatively small central nervous system, some spiders like Portia show complex behavior, including the ability to use a trial and error method. Sense organs. Eyes. Spiders have 4 pairs of eyes on the top front area of the cephalothorax, placed in patterns that vary from family to family. The top pair at the front are pigment carpacelli, also known as little eyes, which in most arthropods are only qualified to detect the path from which light is emanating, using the shadow cast by the walls of the cup. Nevertheless, in spiders, these eyes can form images. The other pairs, called secondary eyes, are believed to be derived from the compound eyes of the ancestral chelicerates, but no longer have the individual facets characteristic of compound eyes. Unlike the principal eyes in many spiders, these secondary eyes catch the light reflected from a reflective tapetum lucidum, and wolf spiders can be spotted by torchlight reflected from the tapeta. On the other hand, jumping spiders' secondary eyes have no tapeta. Another difference between the secondary and the principal eyes is that the secondary has rhabdomers that direct away from incoming light, just like invertebrates. In contrast, the configuration is the opposite in the former. The principal eyes are also the only ones with eye muscles, allowing them to move the retina. Having no muscles, the secondary eyes cannot move. The visual sensitivity of some jumping spiders overreaches by a factor of 10 to that of dragonflies, which have the best vision in the insect kingdom. This sensitivity is performed by a telephotographic series of lenses, a 4-layer retina, and the power to swivel the eyes and integrate pictures from different stages in the scan. The downside is that the scanning and blending techniques are relatively slow. Other senses. As with different arthropods, spiders' cuticles would block out details about the outside world, except that they are penetrated by many sensors or connections from sensors to the nervous system. Spiders and other arthropods have revised their cuticles into intricate collections of sensors. Various touch sensors, mostly bristles called seta, respond to different levels of force, from intense contact to feeble air currents. Chemical detectors supply taste and smell equivalents, often by seta. An adult Araneus may have up to 1,000 such chemosensitive seta, most on the tarsi of the first couple of legs. Males have more chemosensitive hairs on their pedipalps than females. They are responsive to sex pheromones, both contact and airborne, made by females. The jumping spider Evaca culicivora uses the aroma of blood from mammals and other vertebrates, acquired by seizing blood-filled mosquitoes to entice the opposite sex. Because they can tell the sexes apart, it is believed the blood scent is mixed with pheromones. Spiders also have in the joints of their limbs slit sensilla that notice force and vibrations. In web-building spiders, all these mechanical and chemical detectors are more significant than the eyes, while the eyes are crucial to spiders that hunt energetically. Like most arthropods, spiders lack balance and acceleration detectors and depend on their eyes to tell them which way is up. Arthropods' proprioceptors, sensors that register the force exercised by muscles and the degree of flexing in the body and joints, are well understood. On the other hand, little is understood about what other interior detectors spiders or arthropods may have. Some spiders utilize their webs for hearing, where the giant webs serve as extended and reconfigurable acoustic detectors. Locomotion. Each of the 8 legs of a spider is comprised of 7 distinct parts. The part nearest to and attaching the leg to the cephalothorax is the coxa. The next segment is the short trochanter that functions as a hinge for the next long segment, the femur. Next is the spider's knee, the patella, which acts as the hinge for the tibia. The metatarsus is next, and it connects the tibia to the tarsus, which may be considered a foot of sorts. The tarsus ends in a talon of 2 or 3 points, counting on the family to which the spider belongs. Although all arthropods employ muscles connected to the inside of the exoskeleton to flex their limbs, spiders and a few other groups still utilize hydraulic force to spread them. A system inherited from their pre-arthropod ancestors. The only extensor muscles in spider legs are found in the 3 hip joints connecting the coxa and the trochanter. As an effect, a spider with a punctured cephalothorax can't stretch its legs, and the legs of dead spiders curl up. Spiders can develop pressures up to 8 times their resting level to stretch their legs, and jumping spiders can jump up to 50 times their length by abruptly raising the blood pressure in the 3rd or 4th pair of legs. Although more enormous spiders use hydraulics to straighten their legs, unlike smaller jumping spiders, they rely on their flexor muscles to develop the propulsive force for their jumps. Most spiders that hunt actively, rather than relying on webs, have dense tufts of fine hairs between the paired claws at the tips of their legs. These tufts, known as scopulae, are comprised of strands whose ends are split into as many as 1,000 branches and allow spiders with scopulae to walk up vertical glass and upside down on ceilings. Scopulae appear to get their grip from contact with highly delicate layers of water on surfaces. Like most other arachnids, spiders keep at least 4 legs on the surface while walking or running. Silk production. The gut has no appendages except those changed to form 1 to 4, usually 3 sets of short, movable spinnerets emitting silk. Each spinneret has many spigots, each bonded to 1 silk gland. There are at least 6 silk glands, each creating a different kind of silk. Silk is mainly comprised of a protein very similar to that employed in insect silk. It is originally a liquid and solidifies not by divulgence to air, but as a consequence of being drawn out, which alters the internal configuration of the protein. It is similar in tensile power to nylon and biological materials such as chitin, collagen, and cellulose, but is much more flexible. In other words, it can extend much further before cracking or losing shape. Some spiders have a crybellum, a revised spinneret, with up to 40,000 spigots, each producing a single very fine fiber. The fibers are drawn out by the calamistrum, a comb-like set of hairs on the jointed tip of the crybellum, and integrated into a hybrid woolly thread that is very useful in hooking the bristles of insects. Before spiders formed silk coated with sticky droplets, the most primitive spiders had crybella, creating the first silk to capture insects. However, most contemporary groups of spiders have lost the crybellum. Size. Spiders appear in an extensive range of sizes. The smallest patudigua from Colombia are less than 0.37 millimeters in body length. The largest and heaviest spiders occur among tarantulas, which can have body lengths up to 3.5 inches and leg spans up to over 10 inches. Ecology and behavior. Non-predatory feeding. Although spiders are commonly viewed as predatory, the jumping spider Bagheera kiplingi gets over 90% of its nutrition from reasonably solid plant material created by acacias as part of a mutually beneficial association with a species of ant. Juveniles of some spiders in the families Anaphenidae, Corinida, Clubionida, Firmicida, and Salticida graze on plant nectar. Laboratory analyses indicate that they do so purposely and over long periods, and sometimes clean themselves while eating. These spiders also like sugar solutions to plain water, suggesting that they desire nutrients. Since many spiders are nocturnal, the extent of nectar consumption by spiders may have been misjudged. Nectar includes amino acids, lipids, vitamins, and minerals in addition to sugars, and investigations have revealed that other spider species lead lengthier lives when nectar is unrestricted. Feeding on nectar bypasses the perils of fights with prey and the expenses of delivering venom and digestive enzymes. Different species feed on dead arthropods, scavenging web silk, and their shed exoskeletons. Spiders may also eat pollen captured in webs, and examinations have demonstrated that juvenile spiders have a better chance of survival if they have the chance to consume pollen. In captivity, several spider species are also understood to feed on bananas, marmalade, milk, egg yolk, and sausages, capturing prey. The best-known way of game capture is using sticky webs. Variable placement of webs permits different species of spiders to trap various insects in the same place. For example, flat horizontal webs trap bugs that fly up from foliage underneath, while flat vertical webs snare bugs in horizontal flight. Web-building spiders have poor vision, but are particularly sensitive to vibrations. Females of the water spider, Arvioronetta aquatica, construct underwater diving bell webs that they load with oxygen and use for digesting prey, mating, molting, and raising offspring. They live nearly wholly within the bells, peeking out to catch quarry animals that touch the bell or the threads that dock it. A few spiders utilize the surfaces of ponds and lakes as webs, seeing trapped prey by the pulses in the web caused by their struggle. Net-casting spiders weave only small webs that then use them to ensnare prey. Those of the genus Hyptiotes and the family Viridiosomatidae extend their webs and then free them when the game strikes them, but do not energetically move their webs. Those of the family Dinopidae weave even more minor webs, hold them outstretched between their first two pairs of legs, and lunge and drive the webs as much as twice their body length to trap game, and this move may raise the web's area by a factor of up to 10. Investigations have shown that Dinopis spinosus has two distinct methods for trapping prey: backward strikes to catch flying insects, whose vibrations it senses and forward strikes to catch ground-walking game that it sees. These two methods have also been seen in other dinopids. Walking bugs form most of the game of most dinopids, but one population of Dinopis subrufus seems to live primarily on tipulid flies that they catch with the backward strike. Mature female bolus spiders of the Mastophora build webs that are composed of only a single trapeze line, which they patrol. They also create bolus made of a single thread, tipped with a giant ball of wet, adherent silk. They emit chemicals resembling the pheromones of moths, then swing the bolus at them. Although they miss about 50% of strikes, they catch about the same weight of bugs per night as web-weaving spiders of comparable size. The spiders consume the bolus if they have not made a kill in about 30 minutes, nap for a while, and then make new bolus. Juveniles and adult males are much more diminutive and do not make bolus. Instead, they emit different pheromones that attract moth flies and grab them with their front pairs of legs. The primitive Lithistida, also known as the trapdoor spiders of the family Ctenizidae, and many tarantulas, are trap-centric predators that creep in burrows, often shut by trapdoors and frequently covered by networks of silk threads that warn these spiders of the existence of prey. Other ambush predators do without such support, including many crab spiders and a rare species that feed on bees, which see ultraviolet and can adjust their ultraviolet reflectance to match the flowers in which they are stalking. Jumping spiders, wolf spiders, fishing spiders, and some crab spiders catch prey by chasing it and depend primarily on vision to find game. Some jumping spiders of the genus Portia pursue other spiders in ways that seem intellectual, outflanking their targets or drawing them from their webs. Laboratory analyses indicate that Portia's intuitive tactics are only starting points for a trial-and-error approach from which these spiders learn how to overcome new prey species quickly. However, they appear to be rather slow thinkers, which isn't surprising, as their brains are considerably smaller than those of mammalian predators. Ant-mimicking spiders face various challenges. They typically develop slimmer abdomens and false waists in the cephalothorax to simulate the three distinct regions tagmata of an ant's body. They wave the first pair of legs in front of their heads to imitate antennae, which spiders lack, and to hide the fact that they have 8 legs instead of 6. They develop large color patches around one pair of eyes to disguise the fact that they generally have 8 simple eyes, while ants possess 2 compound eyes. They protect their bodies with reflective strands to resemble the shiny bodies of ants. In some spider species, males and females imitate different ant varieties, as female spiders are usually much more significant than males. Ant-mimicking spiders also change their demeanor to resemble the target ant species. For example, many assume a zigzag routine of movement. Ant-mimicking jumping spiders evade jumping, and spiders of the genus Cynomycena walk on the outer fringes of leaves in the same way as pseudomyrmex. Ant imitation in numerous spiders and other arthropods may be for security from predators that hunt by sight, including lizards, birds, and spiders. Nevertheless, many ant-mimicking spiders feed on ants or the ants' livestock, such as aphids. When at rest, the ant-mimicking crab spider Amicia doesn't closely resemble Echophylla, but while hunting, it mimics the manners of a dying ant to draw worker ants. After a kill, some ant-mimicking spiders hold their victims between themselves and large groups of ants to evade being attacked. Defense. There is substantial proof that spiders' coloration is camouflage that helps them to avoid their primary predators, birds, and parasitic wasps, both of which have good color vision. Many spider varieties are colored to merge with their most ordinary backgrounds, and some have destructive coloration, such as stripes and blotches that split up their outlines. Many coloration schemes are present in a ratio that seems to remain constant in a few species, such as the Hawaiian happy-face spider, Theridium grellata. This may make it more challenging for predators to identify the species. Most spiders are weak or unpleasant tasting for warning coloration to offer many benefits. Yet, a few species with potent venom, large jaws, or irritant hairs have patches of warning colors, and some vigorously show these colors when threatened. Many of the family Theraphossidae, which includes baboon spiders and tarantulas, have urticating furs on their abdomens and use their legs to chuck them at aggressors. These bristles are fine seta with fragile bases and a row of barbs on the tip. The barbs generate intense irritation, but there is no proof that they maintain any venom. A few protect themselves from wasps by forming networks of strong threads in their webs, giving the spider time to escape, while the wasps struggle with the obstructions. The golden wheeling spider, Carparethon aureoflava, of the Namibian desert eludes parasitic wasps by flipping onto its side and cartwheeling down sand dunes. Socialization. Several spider varieties that build webs exist in large colonies and exhibit social behavior, although not as intricate as in social bugs. Anelosamus eximius in the family Theridiidae can assemble colonies of up to 50,000 individuals. The genus Anelosamus has a powerful inclination towards sociality. All known American species are social, and species in Madagascar are at least partially social. Members of other species in the same family, but several different genera, have independently evolved social behavior. For example, although Theridion nigroannulatum belongs to a genus with no other social species, T. nigroannulatum constructs colonies that may include several thousand individuals cooperating in game capture and food sharing. Other communal spiders have several Philoponella species, Euloborida, Agelina consociata family Agelenidae, and Malos gregalis family Dictonidae. Social predatory spiders need to protect their prey against kleptoparasites, also known as thieves. More extensive colonies are victorious in this. The herbivorous spider Bagheera kiplingi exists in tiny colonies that help guard eggs and spiderlings. Even widow spiders genus Latrodectus, which are infamously cannibalistic, have begun small colonies in captivity, sharing webs and feeding. In investigations, spider species like Stetoda grossa, Latrodectus hesperus, and Eratigena agrestis stayed away from Myrmica rubra ant colonies. These ants are predators, and the pheromones they discharge for communication significantly deter these spider species. Web types. There is no constant connection between the variety of spiders and the types of webs they create. Species in the same genus can produce very comparable or extremely diverse webs. Nor is there much resemblance between spiders' category and the chemical design of their silks. Concurrent evolution in web building, in other words, the use of similar methods by remotely related species, is teeming. All web designs and the spinning demeanors that create them are the best comprehended. The primary radial then spiral sequence is visible in all webs, and the sense of direction needed to make them could have been inherited from the shared ancestors of most spider groups. Yet, the bulk of spiders builds non-orb webs. It used to be believed that the sticky orb web was an evolutionary innovation, resulting in the diversification of the orbicularia. However, it seems that non-orb spiders are a subgroup that developed from orb-web spiders, and non-orb spiders have over 40% more varieties and are 4 times as plentiful as orb-web spiders. Their more tremendous success could be because sphesid wasps, often the dominant predators of spiders, much prefer to attack spiders with flat webs. Orb. About half the potential game that hit orb webs flee. A web has to perform 3 functions: blocking the prey intersection, sponging its momentum without breaking and stopping, snaring the prey by tangling it or adhering to its retention. No single method is best for all prey. For example, broader spacing of lines will improve the web's area and thus its ability to intercept prey, that decrease its stopping capacity and retention. Tighter spacing, larger sticky droplets, and thicker lines would enhance retention, but would make it more manageable for possible prey to notice and bypass the web, at least during the daytime. Regardless, there are no constant discrepancies between orb webs built during the day and those produced at night. There is no simple relationship between orb web design features and the prey they capture, as each orb weaving species takes a wide range of game. The junctions of orb webs, where the spiders stalk, are frequently above the center, as the spiders can drive downwards quicker than upwards. If there is a clear direction in which the spider can withdraw to avoid its predators, the hub is generally offset towards that direction. Horizontal orb webs are relatively standard, despite being negligibly effective at blocking and controlling prey and more vulnerable to rain and falling debris damage. Mixed investigators have indicated that horizontal webs offer benefits like decreased exposure to wind damage and less visibility to prey flying upwards because of the backlighting from the sky, allowing oscillations to capture bugs in slow horizontal flight. However, there is no one reason for the everyday use of horizontal orb webs. Spiders often link evident silk bands, known as decorations or stabilimenta, to their webs. Field study indicates that webs with additional decorative bands caught more insects per hour. Yet, laboratory analysis revealed that spiders lower the building of these decorations if they discern the existence of predators. There are several unique variants of the orb web, many of them convergently developed, including the extension of lines to the surface of the water, possibly to snare bugs in or on the surface, webs with twigs through their centers, perhaps to conceal the spiders from predators, ladder-like webs that seem most sufficient in capturing moths. Regardless, the importance of many divergences is unclear. For example, the orb-weaving species Sigilla exnotota is known for its characteristic missing sector orb web. The missing sector includes a signal thread to detect prey vibrations on the female's web. In 1973, Skylab 3 took 2 orb web spiders into space to experiment with their web-spinning abilities in zero gravity. At first, both delivered rather sloppy webs that they adjusted fast. Cobweb. Members of the family Theridiidae weave irregular, twisted, three-dimensional webs, popularly known as cobwebs. There seems to be an evolutionary trend toward lowering the amount of sticky silk used, leading to its total lack in some species. The building of cobwebs is less stereotyped than that of orb webs and may take several days. Evolution. Fossil record. Although the fossil record of spiders is considered poor, almost 1,000 varieties have been conveyed from fossils. Because spiders' bodies are pretty soft, most fossil spiders have been discovered preserved in amber. The oldest known amber that contains fossil arthropods dates from 130 million years ago in the early Cretaceous period. In addition to maintaining spiders' anatomy in fine detail, fragments of amber show spiders mating, killing prey, producing silk, and perhaps caring for their young. In several cases, amber has maintained spiders' egg sacs and webs, sometimes with prey connected. The oldest fossil web found so far is 100 million years old. Earlier spider fossils come from a few Lagerstätten, where circumstances were exceptionally suited to maintaining soft tissues. The oldest known exclusively terrestrial arachnid is the trigonotarbid Paleotarbus jerami, from about 420 million years ago in the Silurian era, and had a triangular cephalothorax and segmented abdomen, as well as 8 legs and a pair of pedipalps. Atacopus fimbriomvis, from 386 million years ago in the Devonian period, harbors the earliest known silk-producing spigots and was thus celebrated as a spider at the time of its finding. However, these spigots may have been mounted on the underside of the abdomen instead of on spinnerets, modified appendages whose mobility is essential in web construction. Hence Atacopus and the similar Permian arachnid Permaroction may not have been true spiders and likely used silk for lining nests or producing egg cases rather than for building webs. The most enormous known fossil spider as of 2011 is the Araneidae Nephila jurassica, from about 165 million years ago, recorded from Dowodo, Inner Mongolia in China. Its body length is almost 1 inch. Several Carboniferous spiders were associates of the Mesophilae, a primitive group now represented only by the Liphistidae. The Mesophilid Paleothale monchoensis from the late Carboniferous over 299 million years ago had 5 spinnerets. Although the Permian period 299 to 251 million years ago saw rapid diversification of flying insects, there are very few fossil spiders from this period. The main classes of contemporary spiders, Megalomorphae and Araneomorphae, first appeared in the Triassic well before 200 million years ago. Some Triassic megalomorphs look to be members of the family Caxophalidae, whose current members include the infamous Sydney funnel web spider, and their spinnerets seem adapted for building funnel-shaped webs to capture jumping insects. Araneomorpha accounts for most modern spiders, including those that weave the typical orb-shaped webs. The Jurassic and Cretaceous periods provide many fossil spiders, including examples of many modern families. Arachnophobia. Arachnophobia is a distinct phobia, the unnatural fear of spiders or anything reminiscent of spiders, such as webs or spider-like shapes. It is one of the most typical specific phobias, and some statistics show that 50% of women and 10% of men show symptoms. It may be an exaggerated form of an automatic response that helped early humans survive, or a cultural phenomenon most common in predominantly European societies. What other uses do spiders have? Hungry. Some spiders are used as food. Cooked tarantulas are considered a treat in Cambodia and by the Piero Indians of southern Venezuela. Provided the highly irritant hairs, the spider's central defense system, are removed first. And there you have it. Everything there is to know about spiders, from scientific to disturbing. Join us for another episode of Doc TV's Monster Horror documentary series soon.